Hi, hello. My name is Keiko Hirayama. I'm a developer in scientific content group handling biology and medicine data. In this talk, we are introducing sets of biology and medical related data collections accessible in a Wolfram language and demonstrating how we can access and analyze computational biology data in a Wolfram language. Our curated data collections cover quite diverse areas, ranging from human and animal anatomy to animals and plant species to genes and diseases. And in this Twitch session, we are analyzing and visualizing data using the built-in Wolfram language functions and looking at some examples from each entity domain that showcase the key characteristics of biological systems. I'll go over anatomy and neuroscience, animals, plants, as well as disease areas. And John Cassel, another biology developer, will take over to talk about gene data. So let's start out by looking at anatomy areas. The animal anatomical structure entity domain provides information about more than 90,000 anatomical parts. Here are some sample entities. So anatomical structure like organs, muscles, bones, and body segments, those are known as entities in the Wolfram language. And to find a particular anatomical structure of your interest, you can use control equal and enter free-form linguistics for conversion to inline Wolfram language input. And we have over 130 properties, including image, geometric, and 3D graphics properties to provide detailed information about individual anatomical structures. So for example, you can get information about a bone with a bone's Latin name and get the answer. You can find muscle attachment, joint, or bone articulating information for particular bone. You get the set of properties that describe the bone connections here. And in addition to the textual data, 3D graphics models are available for over 6,000 anatomical parts. So for example, you can ask, show me the 3D graphics of the left hand. And you get a rotatable 3D graphics. And not limited to human anatomy, now we have a new set of animal anatomical structure data available in the latest version 12. The most recent dataset includes computable information about canine and equine anatomical structures. So here are some sample entities. We have dog, liver, muscle, and skeleton here. And like anatomy data for humans, various image and graphics properties are available. For instance, get the typical morphology of the human horse brain, or get the image of the dog's toe bones viewed from different perspectives. And in addition to these graphics properties, we have geometric properties available. For example, you can get mesh region of horse's one bone and compute a mesh area like this. And additionally, you can make use of built-in graphics functions to explore the internal structures of an animal body. In this example, we use anatomy plot 3D function to visualize the joint of a horse. As you can see that meta information is all baked in the model, and you see the associated entities for each subpart. And you can also add some custom stylings here. Here you specify the tissue colors. For example, dogs' muscles should be blue. So here you get the muscles in blue. And there are additional plot themes also available to use to customize your visualization. For example, you get a scientific theme, X-ray theme, vintage themes, and so forth. And in the next example, we feed clip plane options into this anatomy plot 3D function to selectively cut skin and muscles of dog's body to expose the internal structures in the graphics. And with all these accessible tools, you can also visually compare the anatomical parts of an animal with homologous, meaning evolutionarily preserved structures. In the next example, human, dog, and horse feet are displayed in X-ray style, where one of the largest bone, calcaneus bones, is highlighted. And next one involves a bit more coding, but we can also color associated bones and explore the homologous skeletal structures across different organisms. For example, we see that humans have collar bones, while dogs and horse don't. Also, horse has long phalanx bones, which is homologous to the human middle finger bones. And now coming back to human anatomy, anatomy data itself was also expanded since introduced in version 10 and now includes more detailed brain anatomy information. So let's take a brief look at computational neuroscience area. Our collection of 3D anatomy models can illustrate brain's large cortical areas as well as individual gyrus structures and even smaller nuclei. So let's look at the large cortical area. We are looking at prefrontal cortex viewed from different perspectives. And each subpart of the structure can be colored using the anatomy plot 3D function with plot theme, scientific plot theme. You can see the individual gyrus structures. And additional properties help you categorize associated function or identify its hierarchical relationships. For example, here you can see the functions and hierarchical relations of particular brain region. And not limited to individual brain subpart, you can also analyze the components of a nerve pathway. Here you get the components of human visual pathway. And you can get the receptor types, pathway components, direction, and so on. And of course, you can visualize the pathway using anatomy plot 3D. And moving on to the next topic, you can also explore the neuronal network of a human brain. Here, you find the neuronal output of a particular brain region by accessing neuronal output property in anatomical structure entity type. And we can illustrate these output connections of this particular brain region, amygdala in this case, using the nest graph functions. And like this. Also, built-in mathematical graph functionality help us analyze this network further. Feedback loop is one example. It's the key framework in any biological network to fine-tune the system, and you can find such specific circuit that involve particular structure, for example, the amygdala and prefrontal cortex. And you can also explore the optimal flow from amygdala to the output structure, which in this case is a spinal cord, and highlight in the graph. And another thing to note is that you can also identify the individual type of neurons found in the particular brain region. For example, you can find the neurons found in amygdala. And this result returns the list of neuron entities. And further computation allows you to explore their electrical physiological properties. So after looking at components of the brain, let's explore their functions. A variety of cognitive process that takes place in the brain can be categorized and hierarchically mapped. And this anatomical function concept entity, for example, visual face recognition, this describes higher order brain activities and other physiological functions of a human body. And you can find basic information of an entity, including its definition, associated anatomical sites, hierarchical relations, and standard experimental tasks to assess those given cognitive ability. And furthermore, you have an access to statistical maps of brain activity illustrating the brain's cognitive processing ability. For example, find an image horizontally sliced of the part of the brain associated with visual face recognition. This is based on meta-analysis of fMRI studies. And with a very simple line of course, we can also easily create the collage of brain graphics built from front with its coronal, horizontal, and sagittal image slices. So moving on to topics of animals and plants, we have computational data on popular domestic animals. For dog breeds, we have entities describing over 300 different types of popular dog breeds. And you can compute basic information like height, weight, country of origin, or typical temperament for individual type of dog breeds. And you can explore the properties like sorting dog breeds by their height, or and make histogram out of it, or create a word cloud based on dogs' temperaments like this. And most recently, we also introduced set of data on horse breeds. So, for example, saddlebred is one horse breed entity. And for over 240 different types of horse breeds, you can get information like types, use, physical characteristics information. And another thing to note is you can also make use of implicitly defined entity class function to selectively find the group of horse breeds with specific property values of interest. For instance, find the horse breeds used for draft work and endurance riding, or find breeds developed in specific country, in this case in Germany. And another cool thing you can do is analyze and visualize the relations of horse breeds based on their ancestry information and how they are likely developed historically. So you see the relations of horses here. And in addition to livestock breed data, we also have data on species taxonomy. And here are some sample entities. Our database contains taxonomic information for over 1 million organisms, and you can easily feed the taxonomic sequence information to the graph function and visualize the hierarchical organization of species taxa. And the next example, we use graph union function to combine the taxonomic graphs of two separate species and illustrate how they are likely separated in a taxonomic tree. And similarly, plants entity domain provides information about over 170,000 plants. Here are some sample plants entities. And in addition to taxonomic information, you can access their plants distribution information in North America and use geographics function to highlight it on the map. Also, again, implicitly defined entity class function is useful to selectively find plants that have specific characteristics, such as plants with orange flowers and mature height less than 2 feet, and get those plants. And moving on to other life science domains, our disease dataset is recently updated to include additional information about medical conditions and patient statistics. And this domain is closely related to other biology and medical entities. So for instance, you can look up ICD-10 entities, which stands for International Classification of Disease Codes associated with a particular medical condition, or find disease associated with certain anatomical structure entities. And you can also compute a statistical information, compute disease prevalence for a particular country, or compute the trend of diabetes prevalence in the world. And you get the time series data, and that can be fed into the data list plot. And other example here, we get the disease prevalence rate by countries and map them using the geo-region value plot and get the result displayed on the map. So that I will stop here, and John will now talk about gene data.
Hi, everybody. I'll just switch things over here. Good. So I'll be talking about sequence alignment and clustering. In this example, we'll be comparing evolutionary preserved or homologous gene sequences between different organisms using sequence alignment and clustering. We're going to be looking at the EAM1 gene, which has homologs across many species. The Wolfram knowledge base provides homologs for humans, provides sequences for humans, mice, rats, and chimpanzees. For example, here is the EAM1 sequence for mice. So now that we know that what homologs we can use, we can find the entities associated with each of these genes and bring in their gene sequences, of which we'll see the first 5 characters of each. Now, looking at the beginning of these gene sequences, it appears that humans and chimpanzees, once we align them, are quite similar in terms of this gene, while mice and rats are comparatively less similar, but still much more similar than between mice and humans, which are getting to be rather dissimilar. Now, although with that's how it appears from the beginning of the sequences, we should look across the entire sequences to see how similar the genes actually are. So we're going to compute the how similar each of these genes actually are by taking the ratio where we're saying, okay, how similar are each of these two genes normalized by how similar is a gene to itself. From there, we'll be plotting those into a bar chart, and we'll also be seeing how tight these are from a clustering perspective. We're going to be doing that by using turning our similarity ratio into a similarity distance by inverting it here. So this takes a little while to compute, since we're comparing kind of all of the sequences, similarity of all the sequences against each other. Here we go. So as we see, all the genes are, perfectly similar to themselves. Chimpanzees and humans, about 90% similar. Rats and chimpanzees, about half similar, and say humans and mice, about 20% similar. Now we'll also look across the we'll also look across the entire alignment, and we'll see for each section of the alignment how similar they are between each other. So here is predicted humans and chimpanzees cluster quite tightly. Mouse and rats, again, about half. And then here is where the cluster joins overall. So now comparing across the sequence, we observe that compared to the perfectly aligned sequence and the randomly aligned sequence, the sequences fall more or less where we might expect them to, with humans and chimpanzees being very close, mice and rats having, clearly broad areas of similarity, and then mice and humans only having areas that have sort of bursts of similarity between each other. Now, I won't be trying to present the kind of I have also a musical example where we can compare, how similar the alignments sound against each other. I won't try and play it here, but I will kind of show how that was done. So here we'll say if there's a sequence in kind of like the first, if between when we're doing the alignment, if there's only a letter in the first one, we'll play it as a high note. If there's only a sequence of letter in the second alignment, in the second part of the alignment, we'll play it as a low note. And if both are found together, we play both notes at the same time. And between C, G, A, and T, only T doesn't fall on the musical scale, so we map that here to E, forming a nice C major chord. So I'm even though so we can generate these sound sequences, and I encourage you to go to the examples webpage to see to hear that in action. Thank you.